Hello, thank you for having me. My name is Kelsey Wheeler, and I'm a research scientist in the Ribic Lab. And today I'm going to introduce you to our team's mucus-inspired therapies. So let's talk about the most common vaginal condition in women ages 15 to 44, bacterial vaginosis, or BV. BV is a disorder characterized by discomfort. Its symptoms include copious discharge, foul odor, itching, and burning. BV can also lead to serious complications, especially during pregnancy. And right now, 1 in 3 women is struggling with this disorder every single day. At the center of this disorder is a battle between 2 microbes, the beneficial lactobacilli, which are essential for maintaining vaginal health, and the harmful BV pathogen Gardnerella vaginalis. In BV, Gardnerella adheres to vaginal tissues where it secretes toxins and forms thick communities known as biofilms, which are naturally resistant to antibiotics. As a result, antibiotic treatment failure and recurrence are the norm. Nearly 50% of women who are deemed successfully treated for BV will have a recurrence within 3 months, and up to 80% will have a recurrence within 9 months. And BV flare-ups can be a years-long battle for many women. Over time, they might learn what triggers their BV, but because there's no preventative therapies, they're really powerless to do anything about it. As you might expect, women are extremely motivated to find relief, and they often adopt harmful vaginal cleaning practices. And so perhaps it shouldn't have been a surprise that of all of the gynecologists that we talked to, they all said the same thing, that the most urgently needed innovation for this disorder is a safe way to prevent recurrence. Specifically, gynecologists wish they could offer their patients something that could manage BV pathogens without harming the beneficial bacteria or triggering yeast infections, which are 2 common side effects of antibiotics. Now, as it turns out, the body already produces an elegant solution to this problem, and that's mucus. Mucus is a slimy material that forms a protective barrier that's able to accommodate the microbiota while resisting infections. And key to this barrier are the mucins, which are long proteins that are coated with complex sugar structures known as glycans. These glycans have an impressive water-holding capacity, so they're extremely hydrating to the tissues, but they also serve another important function as virulence in attenuating molecules. They're able to tame pathogens that would otherwise cause disease, like Gardnerella, while also supporting beneficial microbes like the lactobacilli. So we were really eager to put these molecules to work against recurrent BV. However, there was an initial challenge. These molecules are incredibly complex, too complex to be produced at scale. So over the last few years, we've figured out a way to reformat mucins into an inexpensive, scalable product. Specifically, we identified the most potent sugars or glycans against BV pathogens and have displayed them on different polymer backbones so that we may deliver these active ingredients and retain them at the host tissue. We call these mucus mimics VRPs, for vaginal restorative polymers, and we've already begun testing them. We've found that our lead VRP is effective against Gardnerella vaginalis, reducing pathogenic biofilm formation by 99%. And at the same time, it's safe for the beneficial lactobacilli and can even boost their growth. We also wanted to test whether mucin sugars are effective against yeast infections. This is important because yeast naturally colonize the vagina, but they are in a harmless yeast form. After antibiotic treatments, they can transition to a harmful filamentous form. Critically, mucin sugars resist this transition and retain yeast in their harmless state. And so in this way, mucin sugars are effective not only against bacterial pathogens, but also fungal ones. So I've shown you that our mucus-inspired VRPs meet each of the 3 design parameters desired by gynecologists. And given the acute need, the lack of preventative therapies, and the prevalence of BVs, we think our VRPs are well poised to make a huge impact in the lives of women suffering recurrent vaginal infections. I'd like to thank the rest of my team, including our faculty lead, Dr. Katerina Rybak, my partner, Dr. Lubov Yakovleva, and our advising clinician, Dr. Caroline Mitchell. I'd also like to thank our funders at the Gates Foundation and the Deshpande Center. We are actively looking to talk with more chemists and physicians as we scale the production of our glycans and our VRPs. And we're also interested in receiving input on relevant animal models and clinical endpoints as we continue testing our VRPs. So I invite you to come stop by our poster to talk more, and I thank you so much for your attention.